Hello friends, I am Anamika Jaiswal from Indore. I am pursuing PhD from DAV and I have experience to 7 to 8 years. Recently, I am working as a professor in at Holkar Science College, Indore. And I have experience also as a coaching school and each and everything. I taught NEET student and 9th 10th student foundation courses, each and everything. I am a biology teacher basically. I taught basic level to advanced level biology. What is biology? You have to understand. Firstly, what is biology? You have to understand. Then you have to clear the concept of biology. Now, shall we start? What is biology? Today I have to discuss about the DNA and DNA replication. What is DNA replication? What is DNA? From onward, you have to clear in your mind what is DNA and what is a DNA replication. Now, shall we start? So now I'm already telling about DNA replication. Now today topic is DNA replication. What is DNA replication? First, we have to understand what is DNA replication. You have to understand DNA replication. You already know what is DNA. DNA is a deoxyribonucleic acid, that is hereditary material. Means each and every living beings have DNA. Some of exception means viruses have exception. Some viruses contain RNA as a genetic material. You know that coronavirus have RNA and that is transmit from one body to another body. That means RNA is a genetic material in only viruses, not other thing. You can say that bacteria, human beings, lower organism to higher organism, everybody contain DNA in their nucleus. So how can we say that if DNA retain inside the nucleus, then what about prokaryotes? Because prokaryote cell that doesn't have any nucleus. So in prokaryotic cell, DNA embedded in cytoplasm, and that space means, suppose that is all our cytoplasm. Suppose I draw the structure of, this is the structure of simply prokaryotic cell, bacteria cell. ठीक है, that is cytoplasm. Here circular DNA is embedded inside here. That part is called nucleoid. Suppose in some bacteria circular DNA embedded here. That part is called nucleoid. Means in this space may be nuclear chromosome here or chromosome here, and that space is called nucleoid, not nucleoid. It is not necessary for the prokaryotic cell DNA retain in the nucleus, but eukaryotic cell, yes, of course, in eukaryotic cell DNA also is must to retain in a nucleus. ठीक है. Now DNA is a hereditary material. Means one parent you can say that one cell is divided into two cells. Means DNA is transmitted from here to here. DNA is transmitted to here. That is the structure of DNA. DNA is basically two types: prokaryotic DNA and eukaryotic DNA. Prokaryotic DNA is circular DNA, eukaryotic DNA is linear DNA. अब eukaryotic DNA is of various type: A, B, C, D, E and Z DNA. Sometimes HDNA is also occur. DNA is double helical structure. In human being, and by Watson and Crick, bDNA was discovered, and that DNA is divided on the basis of their nucleotides. Nucleotides, this friend, means DNA is made up of, first of all, DNA is made up of phosphate group H3PO4, nitrogenous bases and sugar, that all are combined to form nucleotides. Means DNA is a polymer of nucleotides. One nucleotide is combined by another nucleotide by glycosidic bond and form a DNA structure by linear DNA or circular DNA. It depends upon the cell, maybe prokaryotic cell or maybe eukaryotic cell. It depends upon the cell. Which cells are occur and which cell I have to study, prokaryotic cell or eukaryotic cell. It depend upon the types of cell. We have to study prokaryotic cell means we have to study prokaryotic cell means at that prokaryotic cell circular DNA occur, but in eukaryotic cell linear DNA occur. In human being BDNA same as linear DNA is of many types means at one turn. Let me describe how we categorize the eukaryotic DNA AB at one turn. At that one turn, number of bases A, T, G, C, that is nucleotides. That is matlab nitrogen is bases A, T, G, C. There are 4 types of nitrogenous bases: adenine, thymine, guanine, and cytosine. They all are combined to categorized into by purine and pyrimidine. A, T, G, C. That are complementary. Means A always combine with double hydrogen on T and G always combine with triple hydrogen on C. That both the strands are tightly, supercoiled, strongly binded with hydrogen bond, and some other bonds are also involved. Means suppose that is the structure of DNA. Some all bonds are also involved. Hydrophobic interaction, van der Waals force of interaction, all are combined to join and form DNA structure, double helical structure, and that simplified double helical structure given by Watson and Crick. So they in Watson and Crick. So first of all, for the DNA application, many of the enzyme involved. What is enzyme? Enzyme is a biocatalyst, means that is a labor. If you have to prepare the house, suppose we are preparing some house, then what we require? Labor, cement, mitti, int, bricks, all are ingredients. Main ingredients are labor. If you don't have labor, then you don't build up your house, don't build up your building. Same as, suppose some other example, we take some food. We have to chew the food with the help of saliva, but in that saliva there is some enzymes present, that is called salivary amylase, which help in the breakdown of food, and each and every enzyme break its own substrate, not other. Own substrate means suppose nutrients, carbohydrate, protein and lipid. That main 3 ingredients means ingredients in your food which is digestible. Water is also in main ingredient, but it is not digested by any other enzyme. Vitamin is also main component, but not digested by any enzyme. And mineral salt not digested by any enzyme. Only digested nutrient is carbohydrate, proteins and lipid. And these all in nutrients are digested by a specific enzyme. Same here, that hydrogen bond is cut by. If we are not cutting this enzyme, how can we separate? Suppose how can we separate this 2 helical structure? So here suppose A double bond T, that is it. So we have to cut this hydrogen bond, and for this helicase enzyme, first is helicase enzyme. We cut the hydrogen bond, but this is very super coiled. So we have to remove this super coiling. We need required topoisomerase. In same prokaryotic, that topoisomerase is known as gyrase, DNA gyrase. Now that strand will be straight. Na? Then next enzyme is DNA polymerase. Some other thing, the one strand is leading strand, other strand is lagging strand. Leading strand is also called continuous strand. Lagging strand is also called discontinuous strand. But if we have to understand all these things very clearly and so we have to take the time. Not in 5 minutes. We have to take the time, right? We have to clear this all fast now. Now, after that, DNA polymerase for the elongation. But DNA polymerase doesn't have capacity to elongate this chain. So for this required RNA primer, first we have to attach the RNA primer here, and DNA polymerase attach complementary strand. Means suppose at this strand A is A, at this is A, so DNA polymerase apply here T. Suppose G, DNA polymerase apply C. So we have to complementary strand added by DNA polymerase. At that time, suppose here is a final termination point. So, but RNA primer is attached. At that time, DNA polymerase third remove this polymerase. Now again DNA polymerase fill this gap and sealing this process. Is followed by DNA ligase. Then now complete this. That is the DNA replication. थैंक यू फ्रेंड्स, थैंक यू. मुझे पता है डीएनए रेप्लिकेशन इतनी जल्दी 10 मिनट में या 5 मिनट में होना पॉसिबल नहीं है. सो सर प्लीज़ कंसिडर इट. इफ़ I'll join your institute, then definitely student will satisfied. Definitely sir, student will satisfied. डीएनए एप्लीकेशन बहुत डिटेल में बहुत क्लियरली नहीं हो पाया क्योंकि 10 मिनट या 5 मिनट में ये वीडियो पॉसिबल नहीं है सर. इसके लिए पूरी की पूरी क्लास लगती है और डेफिनेटली मैं आपके इंस्टीट्यूट के साथ जुड़ना चाहती हूँ. इस वजह से मैंने ये 5 मिनट में एक शॉर्ट कंसेप्ट दिया है ताकि ये चीज़ें पॉसिबल हो पाए, बच्चों तक डिलीवर हो पाए. और ये तो गारंटी है सर कि मेरे पास बच्चे आके डेफिनेटली मेरे पास बच्चे आके बहुत अच्छे से कंसेप्ट, उनकी सारी चीज़ें जो है ना वो क्लियर कट उनके माइंड में हो जाएगी. आई एम ए प्रोफेसर, बट आई लव टू टीच नीट स्टूडेंट क्योंकि मैंने उनको पढ़ाया है. और जितना मज़ा मुझे नीट के स्टूडेंट या फिर फाउंडेशन स्टूडेंट को पढ़ाने में आता है उतना मज़ा मुझे कॉलेज स्टूडेंट में नहीं आया. और मैं पीएचडी कर रही हूँ सर क्योंकि मुझे मेरे नाम के आगे डॉक्टर लगाना बहुत बहुत पसंद है और मुझे एजुकेशन लाइन से जुड़ना पसंद है. सो गिव मी अपॉर्चुनिटी टू जॉइन विथ योर इंस्टीट्यूट और बायजूस प्लेटफॉर्म टू गिव माई बेस्ट and a student will satisfied. Definitely student will satisfied. Thank you sir.